Organism development, protostomes versus deuterostomes. Animals are classified as multicellular eukaryotic organisms with a few requirements, such as sexual reproduction and using a blastula during embryonic development. They have male and female gametes, which allows for more genetic diversity and thus more resistance to environmental change and disease. Male gametes are the sperm and females are the eggs. When the gametes fuse, they create something called a zygote. This single fertilized egg will eventually become the organism. How does the zygote become an organism? Well, through a four-step process, the zygote will replicate via mitosis until there have been 32 divisions, after which the morula forms. Fluid is secreted from the cells in the morula and create a hollow structure called a blastule, which is important because now the embryos start developing germ layers. Germ layers in animals. Let's begin. Most animals develop 3 germ layers called endoderm, mesoderm, and ectoderm. These animals are called trypoblastic, and tri means 3. Cnidarians or jellyfish only have 2 germ layers since they are less complex. This is called being dipoblastic, and di means 2. The most ancestral animal, phylum, which is porifera or sponges, only has 1 germ layer and is the least complex. Ectoderm. Ecto means outer and derm means skin or covering. Ectoderm is the tissue that will become the outer physical structures of the organism, such as skin, nails, teeth, sweat glands, and even neural tissue or your brain and spinal cord. Mesoderm. Meso means middle. Mesoderm becomes the 3 types of muscular tissue: cardiac, skeletal, and smooth muscle, bones, reproductive organs, and lymphatic tissue or your immune system. Endoderm. Endo means inner. Endoderm becomes the inner linings of the digestive system and the respiratory system. For example, the inner lining of the stomach is made out of endoderm tissue. Scientists like to classify animals into 2 categories based on blastula and germ layer development. The more primitive animals are called protostomes, and as you move down the evolutionary progression, animals are classified as deuterostomes. A protostome almost always has 3 layers. The blastula will develop by creating a mouth first and then tunneling through the gut of the embryo, and this process occurs by splitting the mesoderm tissue to create a cavity within the developing embryo. Protostomes undergo spiral cleavage, meaning that the cells of one pole of the embryo are rotated with respect to the cells of the opposite pole, and they also undergo determinate cleavage, in which the cells are specialized very early in development. Examples of protostomes are sponges, insects, octopi, etc. Now, deuterostomes are a little different in their blastula stage because the anus is going to form first. The hollow cavity of the blastula will be made by using a coelom. They have radial cleavage, where the cleavage's axes are either parallel or perpendicular to the polar axis, resulting in parallel alignment of the cells between the 2 poles. And they also have indeterminate cleavage, where the cells are not specialized until later in development. Examples include sharks, fish, and humans. Why does this matter? Well, these differences in blastula development has led to a great variety of physical diversity among deuterostomes. With having radial and intermediate cleavage, deuterostomes have advanced tremendously in body efficiency, such as the development of the spinal cord. Now, both deuterostomes and protostomes share being trypoblastic and blastula development, but differ in how their cleavage is formed and whether or not the anus or the mouth is formed first. In this video, we have covered gametes, zygotes, blastula development, animal classification, germ layers, and protostomes and deuterostomes. Thanks for watching!